And then at the same time, others can ask questions, and we can learn from there. You can ask your friends questions if you don't understand, or and that's where we learn. And at the end of this session, I would expect that you all combine your informations, your presentations, and that will be your full notes for this drive practical. Okay, so group A, if you're ready, you can start now.
Okay. So maybe I'll start first with everyone can see this, right?
Yes.
All right, thank you. Group A, we're going to start with Wuchereria bancrofti first. So Wuchereria bancrofti or Bancroft filarial worm is a parasitic filarial nematode spread by a mosquito vector. It's one of the three parasites that cause lymphatic filariasis. The other two would be the Brugia. So next we have, this is the adult stage. I'll explain more on this picture. The nuclei doesn't go to the tip of the tail.
Sorry.
So
Can you go back again? Is that an adult stage?
uh, yes, well, more like a stage picture maybe.
Anyone? Any comment whether this is an adult stage? Well, actually, this is not an adult stage. This is um, you can say like a microfilariae. A microfilariae is not an adult stage. The adult stage is not found in the peripheral blood. It's found in the lymphatic system. So this is a microfilariae stage. It's just like a lava or lava on an ova stage, but they are not lava. They are not ova. They are just a specific called a microfilariae stage. Okay.
Okay. So the adult the bodies are usually elongated, cylindrical, got hair-like and slender worm. Body is slightly curved with round ends. They are often creamy white in color, though occasionally their body looks transparent. The body has numerous nuclei dispersed throughout their body cavity, which appear as dark spots on the outer covering. The mouth is very small and devoid of buccal capsule. Male and female is separate and exhibit distinct sexual dimorphism. So from the head end, the cephalic end, both males and females is slightly enlarged and terminates in a slightly round swelling. It has two circles of distinct papillae and is connected to the main body by a short and constricted neck. The tail end, however, uh, lacks any nucleus. Male adults have ventrically curved tail with 15 pairs of minute caudal papillae at the tip of the tail, which are sensory in nature. Out of these, 11 are present in front of the anus and are called preanal papillae, while 4 pairs are located behind the anus and named postanal papillae. On the other hand, on the female tail end, gradually tapers, is rounded at the tip and lacks any papillae. There are no additional sensory structures. So this on A you can see the female and B you can see the male. Uh, in other nematodes, the digestive system and reproductive system of female adults open separately via anus and gonopore, whereas males have a single opening for both systems, which is called cloaca. This is the difference between male and female, where we can see the female is larger than the male. And then this is 100 micrometers approximation, and is 200 to 300 micrometers the width. Posterior end shape curved ventrally and narrowed, while the female is narrow and tapers gradually. Opening at posterior end for male is cloaca, while the female is anus. Reproductive opening open, cloaca and vulva. Then the pineal copulary spicules are absent in female, genital papillae absent in female, and sensory also absent in female. So this is the electron microscopic view of the structure. From A here, we can see that this is the anterior region, the nomatal body showing cuticular striations and sparsely distributed bulges. Large arrow here. So B in this picture here, this is the female posterior end showing an anal opening and also the bulges over here. Then there is C, the surface of the posterior region of female body with cuticular striations and densely distributed bulges. And at D, you can see is the female tip of the female tip with a cuticular wrinkle pattern. All right. This is a area under the microscope. This would be female, the larger one, and then the smaller one would be male. This is the microfilaria stage. Microfilaria of Wuchereria bancrofti are sheathed and measure 240 to 300 micrometer and 275 to 320 in 2% formalin. They have a gently curved body and a tail that is tapered to a point. The nuclear column, the cells that constitute the body of microfilariae, is closely packed. The cells can be visualized individually and do not extend to the tip of the tail. Next, we have the ova stage. The eggs average 40 to 25 micrometer in size. They lack a true shell, instead being enclosed in a membrane which stretches to form a sheath over the embryo. So this is the labeling part. We've seen part of it just now at the anus. And then here's the mouth. So these are the papillae for the male, and these are the female. That has the pharynx, esophagus, nerve ring, gonopore, intestine, ovary, vulva, and then there's anal musculature. So this is the magnification of the microfilariae, 1000 times magnification. This is 400 times magnification. And this is the adult one under the light microscope. So the eggs average 40 to 25 micrometer in size, while the microfilariae averages 290 by 67 micrometer. The adults vary in length from 30 to 100 millimeter, with a diameter ranging from 100 to 300 micrometer, while males are around 40 millimeter by 1 millimeter. So this is the diagnostic stage over here, as you can see from the red arrow. The adults produce the sheath microfilariae that enter the peripheral circulation. Wait. So the microfilariae causes lymphatic filariasis in the blood at night, called nocturnal periodicity. The blood collection should be done at night to coincide with the appearance of microfilariae, and a thick smear should be made and stained with Giemsa or hematoxylin and eosin. Oh, wait. Oops. The microfilariae develop into first stage larva and subsequently into third stage infective larva. The third stage infective larvae may migrate through the hemocoel of the mosquito's proboscis and can infect another human when the mosquito takes a blood meal. The worm infects the lymphatic system in the body and is responsible for removing waste and toxins. If it becomes blocked, then it doesn't properly remove waste. This leads to a backup of lymphatic fluid, which causes swelling. So this explanation is also quite similar to the Bruguier malayi, except that in the Bruguier malayi, the infection happens during the day. So later, this you can also follow this same explanation. So now I'm going to explain the Bruguier malayi. For the adult stage, the size is approximately 32 millimeter for the female, while the 90 millimeter for adult male. Here is the bit of a structure, and then the labeling of the organs as well. Like here's the pharynx, the muscles, the oviductor, and the ovary. It's a bit similar to the Wucherer, Wuchereria. This is the adult stage. You can see here A, B, C, D, E, and F. At A, which is magnification around 2000, there is the endothelial cell. This is the posterior end and the anterior end. Then at B, there's the endothelial cell as well with the posterior end. Then there's the microfilarial sheath at the C, which is 6000 times magnification. The D with 16,000 times magnification. There's the microfilarial sheath. There is the oral ring and the hook as well. Over here, you can see by the arrow. E, which is 15,000 times. You can see the endothelial cell nucleus and then the posterior end. And the last one, F, you can see the hook at 20,000 magnification. So this is the microfilaria just now. Raw magnification 500. This is the size from 175 to 230 micrometer in length. This is the larvae stage. This is length at around 100 micrometer, but the magnification it wasn't stated around here. And here's the larva. Just now there's level 1, 2, and then the infective stage. So the size is around 1,500 to 1,800 micrometer. And this is also the diagnostic stage, which is similar to the Wuchereria just now. So this is the same type of diagnostic stage, which I have explained earlier. So our group A.
Okay, thank you. So actually, there's a error in your presentations. Can you share back your slides, please? Okay, can you go down that slide number 9? Slide number 9.
Okay.
Okay, this one is wrong. This is not belongs to filarial worm. Okay.
Oh.
So this is wrong. This is schistosoma egg. All right.
Uh,
So you can remove these slides later on. Okay.
Right.
Then very important thing is that also there's a mistakes in your presentations that the ova, actually the microfilaria is the ova for this filarial worm. Okay.
Oh.
So because when the adult, it will sheath it will produce egg with the sheath. You can see, can you show me the slides number 11? You can see this one kan. This is actually so-called ova lah for the filarial worm, but we call it a microfilariae. Okay, you can see each of these nucleus of the baby filarial worm lah. Okay, so you can make a correction in your notes nanti. So there's no individual ova for filarial worm. You call it a microfilariae. Okay.
Oh. So there's only microfilary and adult stage lah. 2 stages only
Yes.
adult lah.
Yes. Correct. So what is the key point to differentiate Brugia malayi, microfilariae or Brugia malayi, and Wuchereria?
One of them was infection during the day and night.
No. The characteristic of the microfilariae. How do you differentiate?
There was no nucleus at the tip. For which there is no nucleus, while the Brugia there are nucleus at the tip.
Yes, that's one. Another one is that usually for Wuchereria, you can see the nucleus individually, kan?
Oh, yeah,
From
yeah, yeah.
Brugia, they are very dense. The nucleus are very dense, padat.
I see.
So this how you tell the difference between Wuchereria and Brugia. So let's say in the exam card, if I give this picture, first you look at the microfilariae, the nucleus are all separated, very clear, individual. So that is how you identify um, Wuchereria by looking at this microfilariae. Huh? And if can you show me the microfilariae for the Brugia? Do you have it? On the other slides. I said one. The next slide,
Oh,
19. Here. This is Bulgaria. You can see the nucleus are all padat, right?
yep.
All very, very padat. So this is Bulgaria. So there is a very important characteristic, like one of the important characteristics to differentiate these two types of microfilariae. Okay?
I see. Thank you, doctor.
Good. Well done. Okay. Can we go to the next group, Group B? Group B, are you ready? We cannot hear you. Group B, are you having problem?
Hello, can you hear me?
Yes, yes.
Oh, finally. Wait, I try to share my screen. Can you guys see my screen?
Yes.
Okay.
Yes.
Our group are required. I am Nur Ashikin, and I'm one of the representative of Group B, and there is another representative also. Okay, first, our group are required to search the image of different development, include the adult macrofilariae and ova, to label the parasite, to state the magnification and the size of the adult macrofilariae and the ova, and state the diagnostic stage. Okay, our group B has required to present about the Schistosoma species. I am going to present about the Schistosoma hematobium. Okay, this is the adult stage of the hematobium. There is oral sucker, and here is the ventral sucker. Here we can see the differences between the female and the male hematobium. The female is more slimmer and more longer, while the male is shorter than the female. And next is the Cercaria. This is the Cercaria of the Schistosoma hematobium. This one is the image of the microfilariae, also known as the miracidium, and this is the ova, the image of the ova. This one is the hematobium. There is the oral sucker, ventral sucker, and the females is 20 millimeter long, 0.25 millimeter thick with a cuticular tubercle confined to the end, and there is a sulfuric canal, and male is 10 to 50 millimeter long, 1 millimeter thick, and covered with the finely tuberculated cuticles. Here is a more defined labels of this parasite. Here is the oral sucker, esophagus, acetabulum. This is the male, and this one is the female hematobium. This is the sulfuric canals, and this is the place of the fusion of the crura, ovary, yolk gland, and intestine. This image show the label egg of hematobium. It has a ovate non-articulated. It has with a brownish yellow transparent shell carrying the terminal spine at one point. And magnification and the size of the egg, it is about 120 micrometer in length by 42 micrometer in wide. This is the 100 times magnification of the egg, and this one is the more clearer 400 times magnification. The magnification of the microfilariae is a elliptical free-swimming larvae stage, approximately 200 micrometer long, that are covered with the cilia. We cannot find its magnification. And this is the magnification and the size of the adult. Male is 10 to 50 millimeter long and 1 millimeter thick, and the female is 20 millimeter long and 0.25 millimeter thick. And the diagnostic stage is through the egg from the urine sample. We move on to the Schistosoma mansoni. This is the image of the adult stage of Schistosoma mansoni. This is the macroscopic, and this is the macroscopic. This is the circaria of the Schistosoma mansoni. This is the image of the egg of Schistosoma mansoni. There is a spine right here. And this is the label parasite of the Schistosoma mansoni. There is the darker one is the female, and the lighter one is the male parasite. There is an esophagus right here, uterus, egg, mechanism of gland, ovary, and Kecker junction. And there is also a oral sucker right here, oral sucker, ventral sucker. And this is the size and magnification of the egg of Schistosoma mansoni. The Schistosoma mansoni are large, 114 to 180 micrometer long by 45 to 70 micrometer wide. The size and the magnification of the adult is male worm are robust, tuberculated, and measure 6 to 12 millimeter in length, and female are longer, 7 to 17 millimeter in length. I cannot find the magnification of this microscopic picture. And the diagnostic stage is through the egg and by the stool examination. And next is we move on to the Schistosoma japonicum. And this is the image of the adult stage of the Schistosoma japonicum. The image of the cercaria. During penetration, the cercaria will penetrate without the tail, only the head will penetrate. And this is the image of the ova of the Schistosoma japonicum. This is the level of the parasite. There is a testis, esophagus, the ventral sucker, the oral sucker, the canal, and intestine. And here is also the intestine. This is for the female, and this one is for the male. This is the testis, the non-covulated, and the reunited caca. This is the label of the egg of Schistosoma japonicum. There is a spine right here, but it is difficult to see compared to the Schistosoma mansoni. The spine of the Schistosoma mansoni are much larger than this. And the size and magnification is adult is about 12 millimeter times 0.5 millimeter for male and 20 millimeter for 20 millimeter times 0.4 millimeter for female, and this magnification is 10 times 100 magnification. And the microfilariae is about 200 micrometer times 64 micrometer, and it can be seen under magnification of 400 magnification, but we cannot find the image. And lastly, this is the ova magnification. Its size is approximately 65 micrometer times 50 micrometer, and this is the magnification of 400 magnification. It is the stand with the H and E stand. And the diagnostic stage is the X through the stool examination. Okay, that's all. Thank you.
Thank you, Nurul. So does anyone able to find there are few mistakes there? Anyone managed to identify mistakes just now? There's some error there. Okay. Right. Basically, Nurul, in Group B, there is no microfilariae in schistosoma. Okay. Microfilariae is a special characteristic for the filarial worm. Please take note that for schistosoma, you only have the adult and the larva, which is the cercaria, and the ova stage. Okay, can you go up to your slides? Can you go up a little bit for the schistosoma, the adult stage of the schistosoma hematobium? Just go way up. Okay. Okay, this one is good. Okay. Next, go. The one that shows male and female, slide 6, number 6,
6.
next one. So basically, if you look at this one kan, you know, trematode is when you in block 2, trematode is hermaphroditic. Leaf looks like a leaf. So here, basically, if you look, there are the Schistosoma is the only helminth trematodes. They have male and female separated. Please take note for that one. And also, if you compare to nematode just now, for nematode, female is bigger than the male. But for schistosoma, male is bigger than the female, but the female is longer than the male. So basically, if you look at this male, it has this gynecophoric canal. Okay, the gynecophoric canal is actually for the female to sleep inside and for copulation, untuk mating, for mating. Okay, so imagine in male, for schistosoma, male and female are always together like this, in this form. You can imagine that the male is hugging the female. That's how you imagine how the schistosoma live lah, male and female. Like the male is actually, if you buka the gynecophoric canal, it looks like a leaf actually. If you flatten the male, it looks like a leaf, which is a trematode characteristic lah. But then because the male is so-called hugging the female, and it form a gynecophoric canal. So that's why it looks like cylindrical like that one. Okay. And acetabulum is, if you look at the male, the acetabulum is actually the ventral sucker, also known as the ventral sucker. Okay. So usually, sometimes when we ask question, we can give you like the head of the male, which one is the oral sucker, which is the ventral sucker. How do you want to differentiate male and the female schistosoma? So basically, group B, the adult stage for schistosoma are all the same for all species. Okay, so by right you should have just explain just adult stage for the schistosoma species in general, because for each species the adult stage are more or less the same. The male and the female are look alike. The only difference would be the ova stage. Okay, so you can see for the ova, can you show me the ova for the hematobium? Go to slide 5. This one also can. Okay, so you can see how to differentiate the ova from these different schistosoma species is by looking at the spine, the location of the spine, the shape of the ova, whether it's round, elongated, or whether oval, or then you look at the location of the spine. Okay, for schistosoma, the spine is at the hujung lah, at the end of the ova. Okay, do you have a Mansonia egg? Schistosoma Mansonia. Here. Okay, so if you look at this Schistosoma Mansonia, you can see it has a very large spine at the side of the ova. Just how do you tell the difference? Okay, while if you go for the Japanicum, can you go to the Japanicum's ova? So usually Japanicum, the ova is slightly round, and then the spine are not that visible. Okay, so these are the three common schistosoma that we usually infecting human. You don't have intercalatum. Do you have intercalatum?
No.
Okay, so never mind. You can refer to my note just now. So for schistosoma intercalatum.
Sorry, doctor.
Yep.
The other representative will present it.
Huh? There's another one.
There is another representative for two more species of the cystosoma.
Okay. So the next one, can you go straight to the ova? No need to go to the adult, because the adult are all the same. Okay, next representative.
Okay, good morning, everyone. And our group are going to present about the Cystosoma species, another two species, which is the Cystosoma intercalatum and the Cystosoma mekongii. And this is the macroscopic image of the Cystosoma intercalatum. And this is the ova stage of the Cystosoma intercalatum. Yeah. And about the magnification, the adult stage is about like Doctor said, is same with the Cystosoma species before, so I just skip it. And about the ova stage, the magnification is about 400 times, and the size is about 140 to 240 millimeter, usually longer and more slender than Cystosoma hematobium. And this is the adult stage. Then the diagnostic stage of the Cystosoma intercalatum is the ova stage, and from the stool observation. Yeah. The second one is the Cystosoma mekongii, and this is the macroscopic image of the adult stage of Cystosoma mekongii, and this is the ova stage. Then this is the labeling of the Cystosoma mekongii. This is the intestine, this is the sucker, the testes, the ventral sucker, and this is the ova. About the magnification and size, the adult Cystosoma mekongii can, for male, it can grow up to 2.2 centimeter. For female, it can grow up to 2.6 centimeter. For the ova stage of the Cystosoma mekongii, it can produce 98 eggs per day, and they are usually smaller than Cystosoma japonicum. And the infected person can shed about less than 150 eggs per gram of stool. About the cercaria, on average, only 42 cercaria are released from snail per day. Lastly, this is the diagnostic stage of the Cystosoma mekongii. It is same as the Cystosoma intercalatum, and the diagnostic stage is the ova stage from the stool samples. Yeah, that's all. Thank you.
Okay, thank you. Now it is complete. Okay, so well done. So another important note about schistosoma is that the clinical samples. So again, only hematobium that you collect the urine to search for the ova, while the rest of the schistosoma, you diagnose it from the fecal sample feces. Okay, so well done, Group B. So can we go to Group C?
Okay. Assalamualaikum and very good morning to all. Okay, my name is Balkis. I am represent for Group C. So now we're going to look on Plasmodium falciparum. So let's look on the images of different developmental stage. So first we have ring trophozoite. Okay, as you can see at the above, 3 pictures above, it is the ring trophozoite in a thick blood smear, and another 3 pictures below, as you can see, are also in a thick blood smear. Okay, and for the 2 picture below, we can see that note that the rings can be headphones appearance. Okay, next. Okay, now we're going to look also on the ring trophozoite, but in a thin blood smear. Okay, as you can see, there are double chromatin dot in the infected red blood cells at the top, and also an epilytic form in the infected red blood cell at the bottom. So here we can see the cytoplasm and also the chromatin. Okay, for the other picture also of the ring trophozoite in a thin blood smear. Okay, next. Okay, this one is also a ring trophozoite. Okay, we can see the presence of the Maurus clef, which are often seen in the older ring forms. The Maurus clef stain best with an alkaline of pH of 7.2 to 7.6. Okay.
Sorry, which one is the moral clef? Can you make your slide a little bit bigger, Kak? Too small lah to look at. Okay. Not that big lah. Okay.
Okay.
Yep.
Next one is a mature trophozoite. Okay, these pictures are the first picture is in thick blood smear, and the other two pictures are in a thin blood smear. As you can see, we also can see the chromatin and cytoplasm. So mature trophozoite also can be older mature ring stage. Okay. Next one, we can look on the schizon. As you can see here, there is a mature schizon in the thin blood smear. And for the first picture, you can see a numbers of merozoites and homozoite. And then the second picture, you can see the ruptured schizon where the merozoites are released. And the third picture also a schizon in a thin blood smear. Next one is gametocyte. As you know, the Plasmodium falciparum, the gametocyte look like a banana shape. And for the this picture is in thick blood smear. Okay. This also a gametocyte. Okay, as you can see, the gametocyte on the right is undergoing exfoliation, and this is in a thin blood smear. Okay, next. This is the label of the parasite for Plasmodium. As you can see, they have a nucleus, a plasma membrane, mitochondria, dense granule, ribosomes, apical end, microtubules, and also merozoite coat. Okay. Now I'm going to look on the magnification and the size of the development stages. So here we can use a magnification of 400 times and also 1000 times. For the size, the rings are 1/5 diameter of the red blood cells, that approximately 1.4 microcentimeter. For the mature trophozoite, can be 1.25 to 1.5 micrometer in size. And the schizon 4.5 to 5 microcentimeter, and the gametocyte is 1.5 times larger than the red blood cells, that approximately 10.5 microcentimeter. So for the diagnostic stage is the gametocyte stage. For the peripheral blood film, you can see the banana-shaped gametocyte like I said earlier. And apart from examining the gametocyte, the blood smear also show a high-grade parasitemia. Okay, now we're going to look on Plasmodium ovale. So the developmental stage is here is trophozoite. As you can see, there are vacuole can be seen and chromatin. Also, we can look on the James or snuffer stippling and cytoplasmic ring. So the total magnification we use is 1000 times magnification. So the size of the development stage is smaller than the Plasmodium vivax. That is, the Plasmodium vivax has a small to large size. For the schizon, as you can see in the picture, we can see a dark brown pigment, nucleus, and numbers of merozoite, same as the Plasmodium falciparum. The total magnification is 1000 times, and the size of development is smaller than the Plasmodium vivax that has a larger schizon. Okay, next one is gametocyte. We have male and female gametocyte. As you can see, we also can identify the nucleus in the gametocyte. The total magnification here is 1000 times. So the size of development of the gametocyte is rather like Plasmodium malariae, that is large. For the diagnostic stage is during the schizon stage. For your information, for Plasmodium ovale would be the stippling in all stages except the early ring stage to differentiate them with the other Plasmodium. But as you know, the Plasmodium vivax also have this stippling. So the way we differentiate Plasmodium ovale with Plasmodium vivax would be to investigate on the number of nuclei in affected cells. If the number of nuclei is more than 12, that would be Plasmodium vivax. If less than 12, would be Plasmodium ovale. Okay, that's all. Thank you.
Thank you, Group C. Okay, so just a bit comment. For the Plasmodium falciparum, you failed to mention that for the diagnostic stage on the peripheral blood, the only stage that you will able to see is the ring form, the early trophozoite, which is the ring form stage, and the gametocyte. Because the mature and the schizons are not found in the peripheral blood for Plasmodium falciparum. Okay, it's unlike Plasmodium ovale. You can see all blood stage parasites from early trophozoites to mature trophozoites to schizons and to the gametocytes on the blood films. Okay. Can you share the file, the presentation just now? Just another comment. Can you share back the? Okay, can you show me? Let me see. Go down a little bit. Okay, go down. Okay, stop here. All right. So basically, this is a schematic diagram of a merozoite. It's not a Plasmodium in general. This is a merozoite. A merozoite is what you found it in the schizon. In one schizon, you can find you can see the different number of nucleus kan, and each of these nucleus actually will develop into a merozoite. A baby of the Plasmodium, the daughter Plasmodium. Okay, so once the schizons is ruptured, it will release this merozoite, this particular merozoite, and then this merozoite will invade another new red blood cell. Okay, so just take note that this is not a Plasmodium in general, but it is a merozoite schematic diagram. All right. Another comment is that for Plasmodium falciparum, you say the high-grade parasitemia. So usually for Plasmodium falciparum, the parasitemia is not really high because due to the absence of mature trophozoite in schizons on the peripheral blood, you cannot see it under the microscope. Usually the parasitemia does not actually reflect the parasite count does not actually reflect the actual parasitemia because of the missing of the absence of the mature trophozoite in schizons, because they sequester or they adhere in the blood vessel kan. So because you only see the early ring form and the gametocyte. Okay, so please take note for that one, so you can put it in your notes. Okay, but others are good. Well done.
Doctor, I want to ask regarding for the Plasmodium ovale, through some reference, we only found that usually we used 1000 time magnification. Can it be 400 times?
400 will be slightly
Much
small,
small.
but yeah, but usually for once you usually in the lab kan, when we look, of course, if you look under the microscope, we go for lower magnification first, and then if you want to, let's say we find that one particular parasite, we want to identify it, confirm the identification, then we go to the highest magnification. Usually the 1000 times is the highest magnification using an immersion oil. Okay, then you can see the characteristics of the parasites, not only for ovale but for all of the Plasmodium species. Right. So usually the 1000 times is the best magnification to characterize, to see the detail of the parasites and to identify the different species. Yeah, did I answer your question?
Yes, yes. Thank you, Doctor.
Okay, thank you. Okay, can we go to the group D? Well done, group C. So group D.
Good morning, Doctor. Good morning, everyone. So I'm the one of the representative from Group D, and the other representative will be from the Group 7. So I will start present the class module VVEX. I share screen. Okay, can you see the screen now?
Yes.
Okay, this is the class module VVEX. So these are the pictures, microscopic pictures of different development stages of PVVEX. This is the ring stage. The Plasmodium vivax rings have large chromatin dots, and cytoplasm can become amoeboid as they develop. So these are the image in the thin blood smear. And that is the slight enlargement of infected red blood cell. Okay. This is the trophozoite stages of Plasmodium vivax. It shows the amoeboid cytoplasm, large chromatin dots, and have fine yellowish brown pigment. Juvenile dots may appear more fine in comparison to those seen in the Plasmodium ovale. This is the trophozoites in the thin blood smear. There's the presence of juvenile dot in this picture. Okay, this is the more mature trophozoites. Gametocyte stage, Plasmodium vivax gametocyte are round to oval with scattered brown pigment and may almost fill the red blood cells. This is the Ukinets in the thin blood smears. Okay, next the schizomes. Plasmodium vivax schizomes are large, have 12 to 24 merodoids, yellowish brown and coalesced pigment, and may fill the red blood cells. So these are the schizome in the thick blood smear. You can see the merodoids. Okay. Next is the structure of Plasmodium vivax. We cannot find the specific structure for the Plasmodium vivax. So this is the ultrastructure of the trophozoite of the Plasmodium. So we can see the nucleus and the dense body, mitochondria, and the vacuole, and also the Golgi apparatus, rough endoplasmic reticulum, and the smooth endoplasmic reticulum, and also the free ribosome. So this is the structure of trophozoite of Plasmodium. Okay, this is another picture that we found. Okay, the magnification of Plasmodium vivax used is 1000 times, and these are the size of the Plasmodium vivax. This is the ring stage, and the size of the ring stage is around 2 micrometer, and this is the trophozoite stage. It is around 7 micrometer, and this is the schizome stage. Is around 10 micrometer, and this is the gametocyte stage. Is around 7 micrometer. So the magnification is used is 1000 times. The diagnostic stage for the Plasmodium vivax is ring stage, as it can be detected in the human blood. So these are the life cycle of Plasmodium vivax that we found. So we found that it can be detected in the ring stage in human blood stages. Okay, that's all for the Plasmodium vivax. Thank you. The group 7 will be present for the next Plasmodium.
So where's the other group? The other parasites?
So I'm going to present about the Plasmodium cynomology. So I'm going to share my screen first. Can you guys see my screen now?
Yes.
So good morning, doctor. So I'm going to represent Group G to present about the Plasmodium cynomology. So this is just a very general introduction. This Plasmodium cynomology rarely infect human, and the most cases involve the old world monkeys, such as the baboons and the long-tailed macaques. And the vector is the Anopheles mosquitoes. There are three major development stages, which are the trophozoite, schizone, and the gametocyte. So this is the ring form of the Plasmodium cynomology, and this is the trophozoite and the schizone, and finally is the gametocyte. And I'm going to enlarge a little bit, and I think this one, the darker stained part, is the nucleus or the chromatin, and this one is the cytoplasm, and between the cytoplasm and this nucleus is the vacuole. And the size of the developmental stage is about 5 micrometer, and the diagnostic stage is the gametocyte, and it will be taken by the mosquito during taking the blood meal. And conclusions is usually asymptomatic, naturally acquired Plasmodium cynomology infection, and can occur in humans. I think that's all from me. Thank you.
Thank you, Group D. So basically, for the Plasmodium vivax just now, there's a lot of error in the Plasmodium vivax. Okay, never mind. We go for this one first. Cynomogae, you can see actually for Cynomogae and vivax, the morphology are more or less the same. If under evolutionary, the evolution history of Plasmodium vivax and Plasmodium cynomogae, they are very close related. Okay, and the origins, the history origins of Plasmodium vivax is actually come from the monkey as well, from the primates. Okay, so that's why the vivax cynomogae, their morphology characteristic are almost the same. They have the Schaffner dots. Okay, the Schaffner, the stippling, and then the infected red blood cells are enlarged. Okay, so for the Plasmodium vivax just now, you can see there's an error there because it's very easy to identify Plasmodium vivax. If you look at this image, you can look at the trophozoites, the mature trophozoite. The shapes of the infected red blood cell is amoeboid or irregular. It's not round like a normal red blood cell. Okay, so this is very easy to identify Plasmodium vivax. You cannot miss that. Okay. For Sanomokai, jarang lah. We seldom see it in a human, but it's more or less the same actually, but it's enlarged. Okay. Then for Plasmodium vivax, the very prominent characteristic will be the stippling. The stippling is very obvious. The Schaffner dots, the stippling very obvious until the end of until the gametoside stage. Okay, so you cannot miss that. And there's no band form for Plasmodium vivax. So just now, when your friend shared the vivax presentations, you are actually showing Plasmodium nosai's characteristics, huh? Because the characteristic just now are mostly, I look at it, is you taken from Plasmodium nosai. So please correct again about the slides, the images of the Plasmodium vivax just now, because if you look at this one, this is the real Plasmodium vivax characteristic. You can see the ring form. The early ring forms, yes, it can be looks like a Plasmodium falciparum, but then the vivax tend to the stipplings tend to appear very early at the weak stage as well. Okay, and it's very obvious in the mature trophozoite stage and the schizons and the gametocytes. Okay, so it doesn't have a band form. Please, band form is not a characteristic for Plasmodium vivax. Please take note for that one. Okay, so again, when it comes to the diagnostic stage, these whole stages are all diagnostic stage, from the ring form to the trophozoites to the schizons to the gametocytes. This blood stage, this erythrocytic cycle, the blood stage are all actually at the diagnostic stage. When you do the diagnosis, you look at the stages, you can identify the species. Okay, it's not only the gametocyte, right? And I think another one that is a mistake that done by the Plasmodium vivax journal is about the ookinet. Ookinet is found in the mosquito. Okay, please correct that notes again. Ookinet is found in a mosquito. Anything that's ookinet, the oocysts and everything is found in the mosquito. Okay, very easy to remember. Anything that oo for Plasmodium is found in a mosquito. Very easy to memorize that. Okay. All right. So that is my only comment. So please correct again the mistakes or the error for the Plasmodium vivax just now. Because Plasmodium vivax, the characteristic is enlarged infected red blood cells, stipplings, and the irregular shape. Okay. All right. Group D, thank you so much. Can we go to Group E next?
Hello, doctor. I'm the representative for Plasmodium knowlesi, and the other group will present for Plasmodium malariae. So I'm going to share my screen first. Okay, hello everyone. I'm from PBL Group 9 and I'm going to present about Plasmodium knowlesi. So first of all, the development stages. As you can see in this picture, this is the stages of Plasmodium knowlesi in thin blood smears. So if in the thin blood smear it is clear, that means it is a normal red cell. So from figure 2 and 9, as you can see, is the ring form. So it shows that it is the young trophozoites. From for figure 10 until 12 is the developing trophozoite. Figure 13 until 15 is the mature trophozoite. 16 until 23 is the developing, nearly mature, and mature schizons. And for figure 24 is the mature macrogametocyte for female, and the last one is the mature microgametocyte for male. So next, as you can see here, this is the morphology of Plasmodium knowlesi in a Giemsa stain thin blood smear. So Giemsa stain is very often used to give the parasite a distinctive appearance. So for figure A until F is the trophozoite. Figure G is the schizon, and figure H is the gametocyte. So the scale bar is 5 micrometer. So this is the structure of Plasmodium knowlesi. This is all that we can find, but this is the merozoite, same as the previous Plasmodium presentation. Next is the magnification and size of the development stage. First one is early trophozoite of Plasmodium knowlesi. So all of these is actually the same stages, but shown from different patients. So all of it, for early trophozoite of Plasmodium knowlesi parasite, it is characterized by the appearance of ring-like cytoplasm enclosing a vacuole with a dot of round nuclear chromatin projecting from the cytoplasm. So some of the early trophozoite with small and non-vacuolated cytoplasm appear as a small bluish stained bodies measuring between 1.5 to 2 micrometer. Next is the late and mature trophozoite of Plasmodium knowlesi. So the late, for the late and mature trophozoite, it is characterized by denser cytoplasm compared to ring-like cytoplasm in the early trophozoite. So the cytoplasm of the late trophozoite of Plasmodium knowlesi appeared to be slightly amoeboid and irregular in shape, while the vacuoles were maintained. Late trophozoite measured approximately from approximately 3 to 5 micrometer. And the cytoplasm of the parasite that extended across the erythrocyte, forming the band-like trophozoite that was seen in these 10 patients. Next is the schizons. Schizon stages. Young schizons is characterized by the appearance of between 2 to 5 divided nuclear chromatin masses and abundant pigment granules. And they, for young schizon, they occupy at least 2/3, which is about 5 to 6 micrometer of the infected erythrocytes, while the mature schizons appear to occupy nearly the whole infected erythrocytes. And for gametocytes, it is characterized by their spherical shape, occupying most of the infected erythrocyte. Chromatin was compact and the dark brown pigment was irregularly scattered. Young gametocytes occupied about 2/3 of the infected erythrocytes and were difficult to differentiate from mature trophozoites. And for the last part is the diagnostic stage. So it can be seen by different measures. For the first one is for clinical diagnostic. It is based on the presentation of fever, chills, and rigors. So the clinical symptoms begin after the rupture of the mature schizon infected erythrocytes. So they will release the merozoites and toxic products of parasite's metabolism into the blood. And the rupture of the erythrocytes will release the toxins that will induce the release of cytokines from the macrophage, and it will result in increased body temperature and will lead to fever, chills, and rigors. And next is the peripheral blood film. So in the peripheral blood film, we can see the presence of the ring trophozoite, band form trophozoite, and schizon with no enlargement of red blood cell. But however, the morphology of the Plasmodium knowlesi is similar to Plasmodium malaria and Plasmodium falciparum. So the differentiation is done by the PCR test. Lastly is the parasite count. So the parasite count is using thick film. So the density of the malarial parasites can be read against the leukocytes, and the approximate parasite count can be calculated. So before I forgot that all of the stages can be seen using the blood film that examine in the 1000 magnification and also use the immersion oil. So that's all from me. Thank you.
So someone will be presenting malaria, is it?
Yes, doctor, me. Hold on, let me share my screen. Can you guys see my screen?
Can.
Yes.
Okay. So we will be presenting about Plasmodium malariae. So these are the images of the different developmental stages. So this is the early trophozoite and the late trophozoite. This is the image of the schizon. And then this is the gametocyte. And then this is the schematic diagram of the Plasmodium malariae inside of a red blood cell. So the red color membrane here is the red blood cell membrane, and the parasite is the blue color. And then on the right side, there's a more detailed one with some labels of the parts, like the nucleus, the food vacuole, the hemoron, and the cytostome. Okay, so the magnification and size of the developmental stages. So for the trophozoite, both early and late, this image is taken under 1000 times magnification with oil immersion. The size of the trophozoite are the same as normal red blood cells, or they might be 3/4 the size of a normal red blood cell. For schizons, schizons might have 6 to 12 merozoites with large nuclei clustered around a mass of coarse dark brown pigment. So the merozoites can occasionally be arranged as a rosette pattern. The size is usually between 8 to 12 micrometer. And this image is taken under 1000 times magnification with oil immersion. For gametocytes, the shape is round to oval with scattered brown pigment that fill almost the entirety of the infected red blood cells. So the size is also between 8 to 12 micrometers. And please note that there is no enlargement of the infected red blood cells here. This image is also taken under 1000 times magnification with oil immersion. For the diagnostic stage of Plasmodium malariae, it can be diagnosed at various erythrocytic stages of the parasite development because all of the stages, the ring, the trophozoite, schizons, and gametocytes, can all be observed in the peripheral blood film. But then, in places where humans are in close contact with primates, PCR may be needed to differentiate Plasmodium malariae from Plasmodium knowlesi since their microscopic morphology appear to be identical. That's all from my group. Thank you.
Thank you, Group E. So basically, you cannot differentiate Plasmodium gnocchi and Plasmodium malariae morphologically. So, how should I say this? When you explain Plasmodium gnocchi, you are also explaining Plasmodium malariae morphology. Actually, it's a shortcut way lah. So the first presentation is already briefly mentioned that the morphology of gnocchi and malariae are similar. So the only way is to do PCR and also by looking at the travel history and also the fever pattern and the parasitemia. That's how you tell the difference between malariae and gnocchi. And because gnocchi is tend to have high parasitemia compared to malariae because of the different durations of the erythrocytic cycle. Plasmodium malariae took 72 hours, while Plasmodium gnocchi for 24 hours. Okay, so basically, in general, so the I was quite impressed that for the Plasmodium gnocchi, you're able to find the paper that is published by us malaria team here in UNIMAS, where we study very detailed about the morphology of the Plasmodium gnocchi, which we also mentioned that is very similar, resemble Plasmodium malariae. Okay, so we are done. Group E, right? Now, can we go to Group F? We finished with malaria parasites, so we go to the different parasites. Group F, if you're ready.
All right, Doctor, I will first present about the Trypanosoma cruzi. So is my screen shown?
Yes.
All right, so firstly, I'll talk about the metacyclic trypomastigot. So it's found in the rectum of the triatomine bug, and then it will be excreted by the insect and enter the lesion wound by the bite of the triatomine bug. So this is the metacyclic trypomastigot. So it will be transmitted from the vector to the mammalian host or us as a human. So the magnification is 1,000 times, and the following pictures that I will show are all in 1,000 times magnification. So the size for metacyclic trypomastigot is 12 to 30 micrometer, and this is the infective stage. And later it will transform into the replicative mastigot. So next one. So this is the mastigot, and the size is around 2 to 4 micrometer. So this is a replicative stage in tissue, and then you can see the mastigot here, and this is in the cardiac myocytes. So this is a higher magnification for the mastigot. So you can see this is one mastigot, another mastigot. So this is the nucleus, and this is the kinetoplast. And then this bundle, the mastigot is trying to differentiate or transform into the blood trypomastigot. And then these are pictures of a mastigot in muscle, liver, and spleen cell. So you can see there are bundles of a mastigot in the tissue cells. And you can see like this one is not very clear, but we can see like this is one mastigot, and the biggest one, not biggest, the bigger dot is the nucleus, and the smaller dot is the kinetoplast. And the mastigot is very small, and it's lack of flagella and the undulating membrane. And next one. So after that, the mastigot will transform into the blood trypomastigot, and the size is around 10 to 35 micrometer. And the blood trypomastigot is the diagnostic stage. So it can be taken into the blood smear and seen under the microscope. So the blood trypomastigot can infect other cells and transform into a new cycle of a mastigot. And okay, this all the blood trypomastigot. So they're usually present in like S shape, U shape, C shape, and they have a very obvious kinetoplast at the head here, and then nucleus, flagella, and the undulating membrane. And this one is more obvious, undulating membrane. Right, and then this is the red blood cell. And then this is a more detailed morphology for the trypomastigot. Yeah, so the nucleus, the undulating membrane, flagellum, the kinetoplast, the mitochondrion. All right. So next one, if the trypomastigot in the blood was sucked or ingested by the triatomine bug, and the trypomastigot will further transform into the epimastigot in the midgut of the bug. So the size for epimastigot is 12 to 30 micrometer, and it's the replicative stage in the midgut of triatomine bug. And later it will transform again into the metacyclic trypomastigot in a hindgut. So this one, so the nucleus and kinetoplast of epimastigot is being centralized in the centers, because just now we seen that the trypomastigot, the kinetoplast is here, but for epimastigot it's centralized to here. Okay, so the flagellum, and then the undulating membrane is not obvious in epimastigot, and this one is the dividing form into the metacyclic trypomastigot. You see this one, also the metacyclic form, or not metacyclic, the dividing form. Okay, so this is the general morphology for epimastigot, trypomastigot, and a mastigot. So epimastigot has a more straight and longer body, and Trypomastigot has a slightly slender body, and the kinetoplast is in front here, and a mastigot is a small round cell here. That's all for Trypanosoma cruzi, and my friend will continue about the Trypanosoma brucei.
Okay. Good morning, Doctor. Good morning, everyone. So I'm from Group 11, and I will be presenting Trypanosoma brucei. Hold on, let me share my screen. Are you guys able to see?
Yes.
Okay, so I'll be presenting Trypanosoma brucei. Okay, so first, different development stages. Hold on. Okay, so as you can see here, I will be presenting a little briefly about the life cycle of the Trypanosoma brucei species first, which include the Trypanosoma brucei gabiense and also Trypanosoma brucei rhodesiense. So at first, the safe fly will take a blood meal which injects the metacyclic Trypomastigots. Then the injected metacyclic Trypomastigot will transform into bloodstream Trypomastigots, which are carried to the other sites. Then the next step will be Trypomastigots multiplied by binary fission in various body fluids, such as the blood, lymph, and the spinal fluid. Then next, the circulating Trypomastigots in blood during acute phase will usually be undetectable in latent phase. Then safe fly takes a blood meal again, and the Trypomastigot are ingested. This one is bloodstream Trypomastigot transform into procyclic Trypomastigots in the vector midgut. Then the procyclic Trypomastigot multiply by binary fission again. Procyclic Trypomastigot leave the midgut and transform into epimastigots. And the last stage is the epimastigots multiply in the salivary gland of the safe fly, and they transform into the metacyclic Trypomastigots. So as you can see here, this image depicts the different development stages of the Trypanosoma species in the mammal and also the safe fly. So this one is the bloodstream Trypomastigot, like I mentioned just now, which present in the mammal. Then this one is in the safe fly. This one is procyclic Trypomastigot. This one is long Trypomastigots, which present in the proventriculus of the safe fly. And this one is epimastigot, which present in the proboscis and also the cibarium of the safe fly. And at last, the metacyclic Trypomastigots, which are the infective stage of the Trypanosoma species. Okay, then okay, so this picture shows the procyclic form, which present in the midgut of the safe fly, and it's proliferative. Then it developed into metacyclic form, which present in the salivary glands, and it stopped dividing. Then again, in the mammalian host, the Trypanosoma species is proliferative and is in long, slender form. Then it develops and it becomes short and stumpy form. Okay, so this is a common life cycle, common forms of the Trypomastigot. Of the Trypanosoma species. Okay. So as you can see here, hold on. Okay, so this one is metacyclic Trypomastigots. This form will be injected into the mammal by the safe fly, Then this is the bloodstream tripomastigot that I mentioned earlier. It's transformed from the metacyclic tripomastigot that will circulate and multiply by binary fission in bloodstream extracellularly. Okay, so as you can see here, this tripomastigot, its morphology, the kinetoplast and also the basal body are posterior of the nucleus. So this one is the kinetoplast and this one is the flagella. So this one is nucleus. So the kinetoplast actually located posteriorly to the nucleus. Okay. So the flagellum also form the outer edge of this membrane until the anterior end of the body. Okay. Then next one will be procyclic tripomastigot. Okay, bloodstream tripomastigot ingested by tsetse fly and transformed into procyclic tripomastigot in the midgut of tsetse fly. This one is the epimastigots. Procyclic tripomastigot leave the midgut and transform into epimastigot. As you can see here, the difference between epimastigot and tripomastigot is epimastigot, the size is slightly smaller than tripomastigot, and the nucleus and kinetoplast are both normally centralized within it. Then flagella originate centrally as well. Kinetoplast and basal body lie anterior to the cell body. Yeah. Okay. Next, this one is epimastigot multiply in the salivary gland and transform back into metacyclic tripomastigot. Then later on will be injected into mammals. Okay, so this is the morphological stages of the trypanosoma brucei from the major sites of developmental pathway. Okay. So this is the structure of the trypanosoma brucei. This picture shows the flagellum and the location of nucleus, the kinetoplast, and also some other organelles such as the Golgi apparatus, mitochondrial microtubules, and so on. So I think what's special here is the undulating membrane, which the function is to increase the mobility of the trypanosoma species in the viscous fluid, which in this case is the blood. Okay. Next, this is trypanosoma species under trypanosoma brucei under 400 times magnification. Okay. Then this one is under 1000 magnification and is using Gemsa stain. So tripomastigot length normally range between 14 to 33 micrometer. Okay, so this is the differences between trypanosoma brucei and trypanosoma cruzi. As you can see by differences by the morphology, trypanosoma brucei have a smaller kinetoplast and it divide as tripomastigot in blood. The size is larger than trypanosoma cruzi, which approximately 2 times the size of red blood cell. Then for trypanosoma cruzi, it have larger kinetoplast, divide as emastigot in tissue. Then the size is smaller than T brucei, the approximately size of the red blood cell. Yeah. Okay. So this is the size of the development stages. Trypomastigot is normally 20 to 30 millimeter long, about 3 times the diameter of human erythrocyte, and 2 to 5 micrometer wide. And it's characterized by their wriggling movement. Epimastigot, we can't really find the actual size of epimastigot, but then it's slightly smaller than tripomastigot. So the diagnostic stage of the trypanosoma brucei is tripomastigots. Okay, so this picture shows you the location of the organelle. Okay, I think that's all. Thank you.
Thank you, Group F. Well done. It's very informative, very detailed, and also quite impressed that you can also able to differentiate how the trypomastigotes between T. cruzi and T. brucei. I have no comment because I think everything is presented very nicely, very well. Only a tiny comment if I were you, I would not really focus on the vector side because for your part as a medical doctors in the future, we are interested more on the diagnostic stage, what's in the human, rather than on the vector itself. But it's a good effort, so well done again, right? Great. So the next group, Group G, Leishmania.
Assalamualaikum and good morning to everyone. So I'm Muhammad Izar Akmal from Group 13. So we've divided the tasks in Group 13 to do question 1 and question 3, while Group 14 will do the question 2 and question 4. So for question 1, we're required to search the image of different developmental stages. So next, Ivy. Ivy, can you turn to the next slide? Okay, so Leishmania species is unlike the other hemoflagellates. So what is different from the Trypanosoma species is that it only has two developmental stages, which is the amastigote and the promastigote. So these are the pictures for amastigote. As we can see, there are no like flagellates. We call this as F flagella. So next picture. For this one, we see the promastigote, which contains the flagellum, or we call the flagella. So as we can see, it has like a spine-like shape, or this one. And under the microscope, you can see it has like flagellum, and the amastigote, it doesn't have the flagellum. So then to the size of the Leishmania species. So for the size, we found that amastigote has the shape of a small spherical non-flagellated cell ranging from 2 to 4 micrometer in diameter, while the promastigote is a lens-like in shape and range in size from 4 to 14 micrometer in length, and in width we found it ranging from 1.5 to 3.5 micrometer. So for the magnification, it's found that we examine under the light microscope with high magnification, or 1,000 times, and to identify the Leishmania species. So Group 14 will present the second and the fourth question.
So hello, so before that I'm going to share my screen. It's the same slide as Izzat presented just now. So do you guys see the screen? It's at the second page.
Yes.
Okay. So for the label parasite, for particularly this Leishmania species, there are it's only involved the amastigote and promastigote stages in Leishmania species, where we can see here, this is the the promastigot and the amastigot. The promastigot consists of flagellum, while amastigot doesn't have flagellum, but both of them have kinetoplast and nucleus. So this is the macrophage under the microscopic view under a thousand magnification. Inside the cytoplasm of the macrophage, there's this proliferation of the amastigot, where you can see here. It doesn't seem quite clear, the membrane of the amastigot, but we can see the smaller dark stain inside the macrophage, which is where the smaller one is the kinetoplast of the amastigot, which is in rolled shape like, and then the nucleus is the bigger dark blue stain. So for the promastigot, they have flagellum, and their body are more slender than the amastigot, which is the amastigot are in spherical or ovoid shape, and the kinetoplasts are at the anterior end of the located at the anterior end of the promastigot with the flagellum. So this is the more detailed structure of the leishmania, and with the two stages, which is the only promastigot and amastigot. Essentially, when we see under the microscopic view, we should know and identify the kinetoplast and the nucleus in amastigot, and there's no flagellum. And under promastigot, there will be flagellum, and the nucleus are usually at the central of the body, and the kinetoplasts are located at the anterior end of the promastigot. So the diagnostic stage of the leishmania species is the amastigot, where we can find it in the tissue specimens of the host. So this macrophage microscopic view are from a patient with a visceral leishmaniasis, where we found it from the bone marrow of the patient. So I think that's all for the leishmaniasis species. Thank you.
That's all right. Group G, okay?
Yeah, that's all.
Great. Okay, thank you. Okay, this very simple presentation, yet a very accurate. Just to add to that is that there are many different kinds of leishmaniasis species like Donovani, Brasiliensis, and so on, Mexicana. But in general, the morphology are all the same. So usually, again, the best for diagnosis is by looking at the amastigot stage in the tissue or in the macrophages. Okay, so I think I have no much comment on the leishmaniasis because it's presented well. Okay, good. So we can go to the next one, the last one, group H, Toxoplasma gondii.
Hello, good morning. Can you guys see my screen?
Yes.
Okay. So good morning and Assalamualaikum to everyone. So today I'm from Group H. So we're going to talk about Toxoplasma gondii. So this is the life cycle of Toxoplasma gondii. As we can see here, the definitive host is felids or domestic cats. and the intermediate host can be human, any mammals, and birds. So the first picture here is about techizoid. Techizoid is the, or known as trophozoid. It's tempered anterior and blunt posterior. Tempered mean like the anterior is the anterior end like narrow, and for blunt posterior it's have no shape, like not sharp.
Excuse
And
me.
large nucleus.
Moving,
Sorry?
The slide not moving. Well, I think I'm just still looking at slide 1.
Oh. Wait. How about now? Can you guys see my slide moving or not?
Yes, we can see your slide moving.
Okay, I'll just share like this. So first is techizoid. It's a trophozoid of Toxoplasma gondii, approximately 4 to 8 micrometer long by 2 or 3 micrometer wide, with a tapered anterior and blunt posterior end. A large nucleus, you can see here, and may be found in various sites throughout our body. So this is the techizoid of Toxoplasma gondii in germ cell stance, 1,000 times magnifications. So now is pseudocyst. So pseudocyst can be found during acute infection. Proliferating trophozoid within host cell may appear rounded and enclosed by the host cell membrane. It's called pseudocyst or colony and can be differentiated from tissue cyst by staining reaction, and the rapid proliferating trophozoid in acute infection called techizoids. So this is pseudocyst. Pseudocyst is like it's not really like cyst like that. So for another one is cyst or tissue cyst. Cysts of Toxoplasma gondii usually range in size of 5 to 50 micrometer in diameter. Cysts are usually spherical in the brain, but more elongated in cardiac skeletal muscle, and may be found throughout the body of the host and common in the brain and skeletal and cardiac muscle. So this is a picture of the tissue cyst in the brain. So next one is the same Toxoplasma gondii cyst. So this one, another picture for 1,000 times magnifications. So as we can see here, the cyst, inside the cyst is known as brachiozoids. This using the pus stain. So after that, we can find Toxoplasma gondii unsporulated oocysts. So cysts take about 48 to 72 hours to sporulate and become infective. Matured oocysts measure 10 to 12 micrometer in diameter and contain 2 sporocysts. And then each of the sporocysts contain 4 sporozoids. Human infection may occur either from ingestion of sporulated oocysts or eating uncooked meat, and particularly lamb and pork. So this is the unsporulated oocysts. contain 2 sporocysts and 4 sporozoites. Another picture of unsporulated oocyst. And yeah, these oocysts, unsporulated oocysts, can be found in fecal flotation in the cat fecal feces. So for this is the sporulated oocyst. As we can see here, it's from 2 sporocysts, and then it's contain 4 sporozoites. Yeah. So okay, for now is schizon and microgametes. As we can see, picture A shown intermediate schizon, picture B is matured schizon, while picture C is the longitudinal section of merozoites. Inside of each the schizon, it will become a merozoite later on, and then it will move from the schizon. So schizogony from when the nucleus divides 2 or more times without cytoplasmic division. Merozoite formation is initiated near the center of schizon by development of dome-shaped merozoite enlargement. The merozoite eventually move towards the periphery of the schizon, and then the schizon plasmalemma invaginates around the merozoite, forming plasmalemma of the merozoite. The merozoite separate from the schizon and then leaving the residual body. So here is a microgamon, and B is the zygote. So this is explanation about the microgamon. It's the nucleus moves toward the periphery of the parasites, and then the microgametes is elongated and consists mainly nuclear materials. The microgamons then each of the microgamon have about 21 microgametes, and the microgamete use their flagella to penetrate and fertilize mature macrogametes to form zygotes. So microgametes is the male and macrogametes is the female of Toxoplasma gondii. So question 2 is about the label. So this is the Toxoplasma gondii. Crescent shape with pointed anterior and round posterior. And then the pellicle consists of 3 membranes. This one, 3 membranes, and plasmalemma and 2 closely applied membranes that form an inner membrane complex. So this one is the conoid. It's rotate, tilt, and extend to constitute the host. And this is the roctrice. It is the excretory structure, each consisting of an anterior narrow neck up to 2.5 micrometer long that extend into the interior of the conoid, and sac-like, often labyrinthine posterior end up to 1 micrometer long. So the micronyms are rod-like structure, this small one, mostly at the anterior end of the parasite. It is the specialized secretory organelle. So this is just another picture of Toxoplasma gondii. And as we can see here, the 2 Toxoplasma is surrounded by a membrane called parasitivorous vacuolar membrane, the blue one. So the magnification and the size of development stage. So usually they use 1,000 times magnification, so it can see very clear. So the size of development stages is for tachyzoid, it's about 4 to 7 micrometer long and 2 to 4 micrometer wide. For the bradyzoids, it's about 4 to 8 micrometer long and 2 to 4 micrometer wide. For the oocysts, 9 micrometer long and 14 micrometer wide. For the diagnostic stage, usually for Toxoplasma gondii is tachyzoids. Why? Because it's an actively multiplying form of trophozoite, usually seen in the acute infection. So it can be identified by a crescent shape having a pointed anterior end and a blunt posterior end. Its measure approximately 6 micrometer in length and 2 micrometer in breadth. Contains several dense granules and round nucleus situated between center and posterior end. So they can infect all mammal cell except red blood cell. So this is just another picture of Toxoplasma gondii. Thank you.
Thank you, Group H. I think your slides ada freeze sekejap at the end of your presentation. But never mind. So basically for toxoplasma kan, so usually you will do for diagnosis, it is seldom that we do smear or microscopic examination, except I mean usually we do to determine whether a patient is infected by toxoplasma, we do serology. But then again, we also can do biopsy of the tissue or the infected area. But usually for the brain, toxoplasma is sometimes called like if you look at the YouTube kan, they're referring to like a zombie parasite or something like that because it affects the brain kan, and people with seizures or a different behavior or something like that. Okay, so usually if you want to do microscopy, so usually a biopsy of the brain will tell you about the presence of the parasites on the tissue. Okay, but that is for the presentation just now. It's very detailed, very good. Anyway, and I think we all learned something about the toxoplasma. So well done for the group H. Let me see. Let me share again this. Okay, for those of you who haven't scanned the QR attendance, so here it is. So overall, so today's dry presentation, you've done a very good job for all of the parasites. Again, just to make some correction that I have noted just now, especially for the a little bit on the filaria and the schistosoma, and especially for the malaria parasites. So the malaria parasite, I think there's lots of mistakes there, so you guys can correct that mistakes, and then you can share again with your friends of this practical. While the rest are quite straightforward and very clear. Okay, so before we end these first parts of the practical, anything that you want to, any questions regarding the presentation just now that you're not clear of?
For the time being, everything is clear, Doctor.
Okay. Because because now later on, starting from afternoon kan, there will be lab practical assessment. So the lab practical assessment will be open from today afternoon until tomorrow evening lah, petang. So prior to this lab practical assessment, so you can read back at your notes before you go to your ELIP and answer the EM, because you only have one attempt. Okay. And then I mean all the questions are very simple. It covers the dry practical sessions and also on the wet practical sessions later on. Okay, for the blood smear preparations, I'm going to the lab right now. We will close this Microsoft Teams platform, and we will see you. I will see you again on the other Microsoft Teams platform at 10:30. Okay. So take a break, half an hour, and then I see you again. All right.